Good morning, everyone. And the act of having recorded, you miss these little comments and things and discussion points about a exam on the syllabus scheduled for next week or something. No, we're going to get through this next set of chapters that are focusing on metabolism, and then we're going to do our exam. And yeah, that's all I had to say about that in terms of comments. How are all you fine people? Subdued. Chemistry test this week. Well, I wish I could help you, but if it's just the gas law, I can. But beyond that, no. And I've already explained my feelings about NMR spectra. So, we have finished up our discussion about membranes and ion transport and stuff like that. One of the critical points is that having the plasma membrane being impermeable to ions, and to a variety of different things with charges, we have the ability to generate compartments. And so we've got the ability to have the outside versus the inside. And we're going to, again, we're getting into the idea of how we can make energy from food, how we break down food, and we're going to see that a critical part that was, again, a Nobel Prize winning aha moment is the fact that at the end, the bulk of the ATP energy that we generate from consuming food is generated through the mitochondria, and the mitochondria use a very specific set of compartments to generate a hydrogen ion or proton gradient, and that is what drives the bulk of ATP production. And that's why it's really important to have this previous discussion of membranes before we get into this next step of metabolism. And so that's what we're going to do. I always like having the door open just in case the crowd gets restless. I know I can run and exit really easily. It's just sort of a little thing. All right, so it is probably worth going back and developing a chart that we did early on in the semester in terms of enzyme biochemistry. And it's one of the points I often, at least to myself, think about. The very earliest editions of our text used to include in chapter 3, where we were talking about enzymes, the initial steps in the breakdown of glucose. And it's like, a long enzymatic pathway, lots of different feedback things and substrate interactions, blah, blah, blah. But it hung at the end of the last step, which is what's known as electron transport or oxidative phosphorylation. It's mitochondrial biochemistry, and they just stopped there. I'm like, you brought us all the way 2/3 to the end of the story, and then you're going to stop? And then the story would pick back up in chapter 12 or 13. And, they even would ask me, so what do you think? And I went, probably a better way to put this would be to lump it all together, because what I would do is either jump from chapter 13 and bring it in, or take all this in chapter 3 and say, let's skip this and put it over here. So now what they've done is have this entire section, a couple of chapters, on the generation of energy, both through the mitochondria and the chloroplast. We're going to be talking about photosynthesis as well. And they are very much tied together, as we're about to see here. So we're getting into metabolism, and it's worthwhile to lay out some groundwork because this sort of establishes the rules and the way we can think about the flow. Because one of the things I think is critical in terms of getting this is to think about what's actually happening and to tell the story of the little electron that could, or whatever. So let's begin with that in terms of metabolism. And this is a chart I think you've seen, and that we can break metabolism down into two different sides. We have the building up side, anabolic metabolism, and the breaking down side of catabolic metabolism. And I always keep these two things in mind by thinking about anabolic steroids are what people take when they're roiding up. Anabolic steroids are pushing your biochemistry to build stuff. I mean, the thing with anabolic steroids is if you take them, you don't become the Hulk. You take them and you're able to train harder, recover somewhat faster, get cancer in your later years, but it allows you to begin building greater muscle. So if you don't put the exercise in, you're not going to actually, turn into the Hulk. But, and taking anabolic steroids is just not the way to do it, because you really are setting yourselves up for cancer 20 years down the road. Still, that's anabolic metabolism, is the building up. And so we can define it fairly easily, and as we've mentioned before, it's kind of ping-pong back and forth between these two things. These are opposite sides of the metabolic coin. So anabolic is going from simpler molecules to create the more complex. And so this is all these different kinds of biosynthesis type reactions, the actual transcription, translation, that's all part of anabolic metabolism, the production of proteins, the cell division, all these things that you're building stuff up, making all these complicated biopolymers. The opposite side, catabolic, is the exact opposite. We're going to be going from the more complex to the simpler molecules. And this is what is happening in digestion, breaking down food. As we break down molecules, we're going to be able to get energy out of them, and that's the next thing. From a thermodynamic perspective, this is violating the second law. So we would say this is thermodynamically, A, it is unfavorable, non-spontaneous reactions. B, these are going to require an energy input. That energy input can either be a direct use of ATP, it could be the production of an ion gradient, or using an ion gradient that's existing, whatever. We're going to need to have some kind of energy input. With the catabolic, these are the thermodynamically favorable reactions. Now, we say that they are spontaneous reactions, which of course doesn't mean they happen spontaneously. It's just thermodynamically, we can predict that's the way they're going to go, or they go in the way we predict based on thermodynamics. And that means that these are going to be a net energy release. So we have these two different kinds of biochemical reactions. Those that take energy, those that release energy. We're seeing a little balance going on here. The third part we can bring in is these are typically going to be reduction reactions. That is, we're making bonds. And so, remember, this was part of our definition from the exam last time, how we define reduction. Reduction is a gain of electrons. And so one of the things to keep in mind is that occasionally, in order to carry out a reaction, we're going to need an electron. We're going to need to have an electron being put in. So anabolic reactions often require an electron source in order to build that bond. At the same time, catabolic reactions are typically oxidation reactions. And these don't require oxygen in any way. What they require is a loss of electrons. And as we'll see, pretty much an electron can't go poof into the air. If somebody's going to get reduced, somebody else is going to have to get oxidized pretty much at that moment. If somebody needs to get oxidized, you got to have a place for the electron to go, and so someone's going to have to get reduced. And that brings in our cast of characters. I think of this as sort of a grand musical of metabolism, or opera. Opera would be delightful. I could see someone developing a biochemical opera. It's going to have some sort of music, though, no matter what. And like all good musical opera plays, we have our cast of characters. So let's introduce our cast. And they come out wearing little foam costumes. I'm sure they all have little white gloves. And they're dancing around. Hi, I've never been to Disney World. I've never been on a cruise. But this is what I'm imagining. So we've got a variety of guys that are going to be participating in this reaction. Our initial starting point for today is glucose. So now, contrary, and I still say this, to what my child might say, there are other molecules that you can eat besides glucose or glucose polymers. We don't live on carbohydrates. We'll get into how you actually do break down proteins and fats in a bit, but this is really where we do start. So, okay, glucose. And we know from a physiological perspective, you've got to maintain a good blood glucose level. If your blood glucose level goes down to 0, heck, if it goes below 70, that is, as we say, not compatible with life, i.e., you'll be dead. So we maintain a high level of glucose in the blood. Of course, if it gets too high, that's diabetes, and you end up with a whole set of problems. But your brain is running on glucose. Glucose, just to remind us, this is a 6-carbon sugar. And we can extract glucose from a bunch of things. All starches are pretty much glucose polymers. We break them apart. Your liver stores glucose as glycogen, which is kind of its own little starch, a little bit shorter chain. You break that down, dump glucose in the blood like that with adrenaline. If it is some kind of sugar, not glucose, it probably can be isomerized, turned into glucose, and broken down that way. Glucose, you can guarantee that's good fuel. If you need your cells to grow, you give them a little bit of glucose and pat them on the head and say, go have your pixie stick. Because glucose is always, pretty much always, a good food to have. A thought? No. Okay. You just want some glucose. So have your pixie stick. We should all be doing that. All right. 6-carbon sugar. The 6-carbon part is important because we're going to be counting carbons. Now, a couple other members of the cast that we need to keep in mind. Let us never forget our dear friend adenosine triphosphate, ATP. And this contains a phosphate. And of course, whenever we say phosphate, what do you say to yourself?
Negative charge.
Thank you, Hannah. Everybody should have been hollering negative charge. We should stand up and holler negative. All right. So the phosphate on ATP is what's driving all these different anabolic reactions. You can either covalently attach that phosphate to an aspartic acid in the sodium potassium ATPase and change the shape. You can have that phosphate being broken off in a little pocket, and that negative charge is hanging there. It's not being covalently attached. That's going to have a change of shape of proteins. This is what's driving protein shape change, which in turn is making stuff happen. And one of the things sort of in a meta is as I'm doing this, I'm starting to think about the fact that my muscles are in fact breaking ATP while I'm talking about the ATP hydrolysis. How amazing is that? So ATP is our number one energy source. And just from a nerd perspective, there are occasions where we will be seeing not just the adenosine triphosphate, but we will make a guanine triphosphate. GTP, while it's not the same as ATP, all of our nucleotide triphosphates have the ability to serve as phosphate donors. These are all, by the way, ribonucleotides. They're not deoxys. All the ribonucleotides, they actually can they can participate in different kind of phosphate donor reactions. And so, off the top of my head, we know ATP. We can put in a lot of different things. UTP, uridine triphosphate, actually is used as a phosphate donor. And so we have uridine triphosphate becoming uridine diphosphate, that is uracil. Guanine is very definitely doing this. So we have a collection of proteins we'll be getting into called G proteins. They bind GTP. And so we've got a whole bunch of GTP binding proteins. Cytosine triphosphate, as I go through this, I'm thinking, I can't think of an example where CTP is being used as a phosphate donor. And it's one of those things, I'll have to go look that up. I don't know. I never hear CTP being used, whatever. So there's going to be some points where we're going to lump together the triphosphates and just say, yeah, it's ATP. Okay? Just so you can be clear. We're going to have our grand tote board for the amount of nucleotide triphosphate that we generate. And we're going to put together occasionally a GTP in there. So let's not be pedantic and all, oh, that's not ATP. Yeah, I know it. Nucleotide triphosphates. The main point to make here is 1 glucose in the presence of oxygen is going to give rise to around, just the number I'm going to use, 36 ATP. Okay? So this is a way of thinking about how many ATP we can get per glucose. It's another one of these things to be keeping an eye out for. It's kind of like, a checklist that we can see where we are in the process because we know what we should be getting. And again, this number, depending on how wonky you want to get, we can get way in the weeds, 32, but then there's this, well, whatever. I'm sticking with 36. Bite me on YouTube. Put it in the comments. All right. Now, there are 2 other main players in this game. And these are our electron acceptors. And so, we had the spotlights come down and say, meet your electron acceptors. And we've got 2 main electron acceptors. One is number 1, my favorite electron acceptor, and that is a molecule known as nicotinamide. Oh, heck, I can't spell it. Okay. Nicotinamide adenine dinucleotide. And so he, in fact, is based on a nucleotide structure. We refer to my friend nicotinamide adenine dinucleotide. You know him as a niacin derivative, as a B vitamin. We refer to him as NAD. And I prefer that you call it NAD and not NAD. I can't go there. Now, this is an electron acceptor. Consequently, it's going to have multiple forms. It can be reduced, that is, it's picked up an electron, or it can be oxidized. It's dumped an electron. This is the form we write it in when it's in the oxidized state. It's ready to pick up an electron. So when we write it as NAD, this is the oxidized form. We have a shorthand for when it picks up an electron. When it has been reduced, it is written as NADH. That is the reduced form. Now, another electron carrier is a guy that we only see limited. As you get further into biochemistry, there are other roles for this guy, but frankly, I'm like, why are you even here? You're not doing me any good. Get the hell out. But no, it's like, I want to play. Okay. And it is another B vitamin type. It is called flavine adenine dinucleotide. And he goes by the name of FAD. Again, FAD is how we write him in the oxidized state. When it can pick up an electron, it is written as FADH2. So, if you see FAD, that means that it can pick up an electron. If you see FADH2, that means it's done picked up its electron. Now, another thing to keep in mind with this is this system is very much of a internal recycling system. And There's a little memo up there on my screen that's bugging me. Sorry. All right. So the inputs are glucose and oxygen. That's stuff that you have to actually get from your external environment. All right. You got to breathe. You got to eat something. All the rest of these things are in the cell, and there's not new stuff coming in. There's not stuff going out. In that, a cell has got a supply of electron acceptors, NAD and FAD. It's got a supply of ATP. And this stuff in the cell is either in the triphosphate form or the diphosphate form. It's either in the oxidized form or the reduced form. You're not going to add anything to the cell. I mean, they sell supplements for NAD and FADH2. You can find these things. I mean, total hooey. Because every cell has got the amount that it is able to use at any one point. And putting more in, there's nothing for them to do. And so it is a grand recycling system. And that's really what we're looking at is the state of these guys. You're either, with the nucleotides, you're either in the triphosphate or you're in the diphosphate. With the electron acceptors, you've either picked up an electron or you're ready to donate an electron. I can't remember if I said this little factoid, but I love this notion that an ATP molecule in your average cell, pick a cell, any cell, that ATP molecule is going through 500 phosphorylation, dephosphorylation, phosphorylation events every single day. And you generate your body weight in ATP every day. Now, of course, you're not consuming that. You've got this finite amount of it. And what you're doing is going from ADP, rephosphorylating it to ATP. So that's a really important thing to keep in mind of how this game is working, is we got these inputs, glucose and oxygen. Everything else is just this gigantic wheel that we're turning, and you're going one direction or the other. Now, you know, a grand toast might be, may all your nucleotides be in the triphosphate state. Because if everything you got is in the triphosphate form, well, A, you don't need to eat because you got ATP. B, what the hell are you doing that you're not breaking apart ATP into ADP? You aren't doing anything either. So in order to carry out this thing we call life, you've got to constantly be phosphorylating ADP and dephosphorylating it. One final point, and this is like I'm starting to write in candidacy or a Facebook page, I should. We talk about ATP synthesis. And, you know, you do actually synthesize nucleotides, something called the pentose phosphate pathway. Go for it, man, when you get to biochemistry. That's how you actually biosynthesize nucleotides. We're not talking about ATP synthesis in this. We're talking about ADP rephosphorylation. And it aggravates the heck out of me that we talk about an enzyme called ATP synthase. Like, dude, it is ADP rephosphorylase. These are the small things that keep me up at night. Okay, I'm better. So this is pretty much our cast. And what we're going to see is how these guys bounce back and forth between their two states. So we have ATP, and it can be broken down to ADP, and a little shorthand term you'll see. And some of these things are things I've been seeing for an embarrassingly long time and wasn't quite sure what the hell they meant. And I'm passing that on to you. So you have, we write it as ADP plus a P with an I on it, or probably it's an I if you're IP or pi. Inorganic phosphate is what's being released. Just as a little note, there are occasions, not in our metabolism, but there are occasions where we will go from an ATP and generate a monophosphate, like when you're incorporating nucleotides into a growing nucleotide chain, you pull off two phosphates, not one. And the two phosphates are known as pyrophosphate. Why don't they call it diphosphate, man? I don't know. There is a whole form of DNA sequencing that, I don't know how it's probably not practiced much, but it's known as pyrophosphate sequencing, is that you can detect if a nucleotide has been added to a growing nucleotide chain by the release of pyrophosphate. Just a little trivial point. There you go. So these are our characters. Now what we're going to do is begin putting them together into this grand scheme and start generating our sought-after 36 ATP. Let us begin. So, if you look in your book, there's a couple of figures at the very beginning that talk about metabolism in this really grand, big way. And it's worth probably a quick graph here. You recall back when we talked about enzyme biochemistry, this kind of energy graph. This is how we introduce the notion of the activation energy. And so we've got just relative energy of the molecules here and the reaction progress here on the x-axis. And we generally would put up glucose, and if you're being careful, you put in oxygen. And we go down, and the products are going to be carbon dioxide and water. Are you in TV land able to see that? Kind of. You could come to class. There's only 7 people here. We're glad to have you. 5 people here. All right. So these are the products. And just as a little thought, anything down on this side of the curve is going to be a good fire putter outer. Anything on this side of the curve ain't a good fire putter outer. So you probably don't want to try and put your fire out by spraying pure oxygen on it. Not going to work real well. Again, if you have a fire in the kitchen, don't take powdered sugar and begin throwing it on there. That might not work. Just saying. Carbon dioxide, water, those are like good fire extinguishers. All right, so that's what we're looking at. Now, we know then what is released is energy. And, there's a fair amount of energy in a mix of glucose and oxygen. So this energy, not inconsequentially, this is what happens when you burn stuff. This is combustion. If you just set this on fire, you get this big release of energy as heat. And that's not going to work for us for a lot of reasons we've talked about, one of them being that level of temperature denatures our proteins, so we can't do that. Second, we need to have a storage spot. I mean, if we had like a boiler where you could heat the water up and then use the steam, there's a lot of mechanisms. What we're going to do is extract that energy and do a little bit of process towards making ATP. But we're not going to be able to use that energy in one reaction. Consequently, in order to carry this out, we're going to have a bunch of small reactions. And this bunch is, in fact, what makes biochemistry interesting. Quote. The audience seems to be stable. I'm not fearing for my life that I have to run out, so I'm shutting the door. So, rather than having one reaction, whoosh, and all this energy coming out, like setting fire to something, we're going to have a whole set of reactions. Each one has its own little bit of activation energy, because we've neglected to put that in. But we know there's an activation energy of the heat of a match. And they say a match, when you strike it, it's hitting like 700 degrees for a few moments. 450 is enough to set this thing off. So you have a fair amount of heat you got to do to get the whole thing to go in one throw. But by breaking it apart, we can do it at body temperature and release small amounts of energy we can capture. All right, so let's now jump into the process. We start out with glucose, no oxygen required, and the first step is one of the most evolutionarily conserved, primitive, first on the scene type biochemical reactions. And that is the splitting of sugar known as glycolysis. Glycolysis. Glycolysis, it's been discovered, the pathway's been known for quite a while, probably like in the 1880s or so, but I just made that number up. But glycolysis is something that pretty much every cell that you can find is carrying out. So, an elephant and a bacteria have a date in a bar. They can sit and talk about glycolysis. They may not have a whole lot of other things in common, but we always can talk about glycolysis with each other. All of our cellular friends, not just bacteria, not just eukaryotes, no. All of us can talk glycolysis. We should talk glycolysis with our bacteria friends. This is where we begin. And it starts out like this. We've got the 6-carbon glucose. And just as a little point here, the level of what you need to know is pretty much what goes in and what comes out. When you take biochemistry next semester, you can memorize and learn every single 6-carbon and 3-carbon structure to your heart's content. For our purposes, you don't need to know the structures. Don't worry about it. Getting the flow is the critical thing. So, the 6-carbon glucose. The first thing we're going to do is actually add some phosphates from 2 ATPs. So we've got 2 ATPs back up onto this glucose and dump their phosphates, and they go out as 2 ADPs. We now have glucose bisphosphate. But don't worry about writing that down. It immediately splits into 2 3-carbon molecules. So glucose now is going to split into 2 3-carbon molecules. And for you biochemical nerds out there, they are initially not identical 3-carbon molecules. This is going to be phosphoglyceraldehyde. There's one of them is phosphoglyceraldehyde. The other one is a different 3-carbon molecule. There's an isomerization reaction that makes it into the same. You don't care. The point is, we now have these reactions going on in parallel. So, the next thing we do is beginning to actually oxidize this guy. So, we're going to oxidize it. That means we have to have one of our electron acceptors. Here comes NAD, ready to pick up the electron. NAD comes in, beep, 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 picks up an electron. I think of them as little dump trucks. NADH is what is now coming out. Same thing happening here. NAD, NADH. Very good. We still have 3 carbons on both these guys. The next step, finally, we're going to start extracting a little bit of energy in the form of ATP. In that, we've got, for each one of these guys, 2 ADP can back up and use a bit of the energy and phosphate floating around to generate the triphosphate. Same thing happening here. And we got 2 ATP here. So a grand total of 4 ATP have been generated. Another little nerd point here, 2 points. This is a distinctly different way of making or adding the phosphates, of rephosphorylating ADP, than the bulk of how we do it. And this is referred to as, it's known as substrate level phosphorylation. And I only bring this up because it is a distinctly different way of making ATP than the end game, which is what's called oxidative phosphorylation. And this, again, is a really, really old evolutionary way of making ATP. All right, so we are now nearing the end of this line, and I'm running out of space. We have still 2 3-carbon molecules. We haven't really talked about the names of these different guys as they've been turned around, but let's go ahead and name this guy. These 2 3-carbon molecules that we finish with glycolysis are known as pyruvic acid. Also in the biz, referred to as pyruvate. Just depends on the pH. So, we now have 2 3-carbon pyruvates. All right, let's go to our tote board and think about what's going in and what's coming out. So, in glycolysis, what goes in is 1 6-carbon glucose. We have to have 2 ATP to start this thing up. And we need to have 2 of these oxidized NADs. So that's what's going in. Now, if you wanted to be really picky about it, you're also going to need 4 ADPs floating around. But with that specific molecule, with the nucleotide triphosphate, we don't have to worry about that being around. As I said, if all of your nucleotides are in the triphosphate form, you're not carrying out this reaction. So having ADP around is always around. And so I don't put that into the tote board. Sorry. That means what comes out, because this is not critical to the story, is 2 3-carbon pyruvates, a grand total of 4 ATPs, 2 here, 2 there, and we now have 2 NADHs. So, a couple of things to look at. I was told one glucose molecule was going to get me 36 ATPs. Where are my ATPs, man? Because by my calculations, if you put 2 in, you get 4 out. That's a net gain of 2 ATP. And you're telling me I'm supposed to have 36? I'm feeling a little bit shortchanged here. What the heck? Additionally, we've got this pyruvate molecule. The other thing about this was in our chart, we ended up with carbon dioxide. We haven't kicked out any carbon dioxide yet. So clearly, we have only just begun our pathway here. But this is, a start. We have begun to break down glucose. This is glycolysis. Another little interesting nerd point, just as a moment. I will take a theoretical sip of coffee. I don't have any coffee, and so I'm sad. Have you ever heard of the supplement called creatine? And since this is all non-male bodybuilder types, anybody know creatine? Have you ever taken it?
Mm-hmm.
You have?
Yeah.
For what?
I was doing weight training for a while.
Cool.
So vitamins.
Did it do you any good? Yeah, it's sold as a supplement. Did it do anything for you?
Not really.
Yeah, all right, there are people that will say, oh, no, I take it. Creatine is part of this pathway. It's not exactly, but creatine is an ATP regenerating system. There's an enzyme called creatine phosphokinase that is involved in substrate level phosphorylation of ATP. The idea behind taking creatine is when you're carrying out metabolism that is requiring a lot of ATP generation, like heavy muscle lifting, and not doing it over a long term like a marathon, but doing it really intensely, somewhat anaerobically, as we'll see. If you were lacking in some of these molecules, it might help. But you're not. Pretty much study after study has shown pretty unequivocally that vitamins taken externally, as a pill, don't do much for you. Supplements are not really a great benefit. Creatine, if you go looking around, you got to dig pretty hard to find anybody that says creatine actually has a significant effect on you. What it does have a significant effect on is the bottom line for General Nutrition Center. That's what it is. It's for sale. And I am just blown away, find it delightfully hilarious that study after study, year after year, will come out and say, supplements really don't do much for you. And General Nutrition Center, they could put a banner up there. It's GNC. What we sell doesn't do jack for you. And it wouldn't have effect into their bottom line. And so we go into the GNC at the mall, and I'm being a smug and snarky biochemist, and I sit there and look at these things going, oh, give me a break. Oh, no, really? Oh, my God. Sorry, GNC. God, and now this is going to be publicly put out. I speak only for myself, not for the university. But I have GNC stock. No. Vitamins. And, a lot of these guys are in vitamins. That's what, niacin and flavonoids and things are part of this stuff. We'll see with like B12, part of this stuff too. You got to be pretty screwed up if you need to take external B12 and these things. Just saying. All right. So that is what goes in, what comes out. And in fact, that's really all you need to know when we say, okay, tell me about glycolysis. What goes in is 6-glucose, 2 ATP to start it up. You're going to need 2 NAD. What comes out is 2 3-carbon pyruvates, a net gain of 2 ATPs and 2 NADH. Thank you very much. Next question. The next step is another really cool biochemical pathway. Glycolysis was discovered very early on, and it's one of these things that I say to myself, I should go look up on Wikipedia. I say it was discovered back in the 1800s. I expect it is. The next step requires a bit more thought and actually some fairly brilliant organic chemistry thinking. And it doesn't happen until the period between World War I and World War II. And this is what's known as the Krebs cycle. And it's our next step. We're only going to just introduce it for a moment, and then we're going to have to break, but there you go. So we've got the Krebs cycle, named for Hans Krebs. It's also, and it must be because sometimes people don't like Krebs. He wasn't the nicest guy. I don't know. I've never met him. Krebs was working with a guy named Otto Warburg, who had developed something that is known as the Warburg manometer. And it looks like a giant crack pipe. And it's a way of very carefully monitoring the carbon dioxide evolution of a chunk of tissue. And they would take pieces of liver from some living creature, presumably not one of the lab technicians or something, a rabbit, and you stick it in there, and you would put different metabolites, and as it was carrying out respiration, it would give off carbon dioxide. And they were monitoring this carbon dioxide. And so you have the tissue over here, and then this very long pipe with a very skinny bore that the bubbles can come through, and then you can see where how much bubbles are being generated by looking at these different marks on one side of the tube. And so the manometer is placed into a 37-degree water bath that can be maintained at temperature, and a big manometer might have like a dozen of these individual tubes in this great big metal water bath, and it's quite the thing. Apparently, Krebs fled Germany after World War I to the UK, and he brought with him some of these very precious manometers, and he began carrying out experiments on looking at what was going on the next step. And part of what makes this kind of like, when you think about it, it's like, damn, that is some serious out-of-the-box thinking. This is a linear kind of breakdown. And it's what referred to at one point like the mountain flow or mountain stream hypothesis, where you start up high and you go down. Krebs assumed that the breakdown of pyruvate would be the same thing, but oh no. The Krebs cycle is a good bit more complicated. The Krebs cycle is also known as the tricarboxylic acid cycle, also abbreviated as the TCA cycle, also known as the citric acid cycle. And there was a time where I'm thinking TCA stood for the citric acid cycle. It doesn't. It's a tricarboxylic acid cycle. I'm going to give Krebs his due. I always call it the Krebs cycle. So, don't forget to support us on PayPal. The Krebs family is whatever. Bye-bye.